In this video, we will take some time to break down two of the four main macromolecules, carbohydrates and lipids. Let's start with carbohydrates. When you hear this term carbohydrate, the first thing that should pop into your head is sugar, because carbohydrates are nothing more than sugar molecules either seen individually or linked together in a chain. The basic subunit of a carbohydrate is a monosaccharide. Mono prefix meaning one, and saccharide suffix relating to sugar. So a single sugar molecule is a monosaccharide. And an easy example to remember for this is glucose. This is an important one to know for many reasons, especially when talking about how cells obtain energy through breaking down glucose molecules like this. But more on that later. But sugar molecules are not only seen as individual molecules, they can be linked together to form larger carbohydrate molecules like disaccharides and polysaccharides. Di meaning two and poly referring to many or more than two sugar units in a chain. Our example of a disaccharide here is sucrose, which is when we have a glucose molecule and a fructose molecule linked together with a chemical bond. In order for this link to occur, a condensation reaction takes place, meaning two hydrogen atoms and one oxygen atom leave during the formation of the bond, which forms water. This formation of water here is what gives it the condensation reaction name. But there is another name for this bond, which is a glycosidic bond. A glycosidic bond forms when a sugar molecule chemically bonds to another group, which in our case is another form of sugar, making this a larger carbohydrate. These links can continue to add more sugar molecules to the chain, forming a larger polysaccharide structure, which we see here as amylose. Long-chain polysaccharides are commonly found in biological systems, and we need to know their structure and also what types of organisms make them. There are three broad categories of large carbohydrate molecules, which are starch, glycogen, and cellulose. Starch and cellulose are only made in plants, while glycogen is made in animals. And looking at a few structural differences, we can see that cellulose structures are unbranched, starch structures are moderately branched, and glycogen is heavily branched. And if we look at the similarities between these three molecules, we can see that all of them are made out of the same repeated structure, glucose. But before we talk more about these larger molecules, let's talk a little more about the structure of glucose. Glucose is a single sugar molecule that has a chemical formula C6H12O6, and commonly, but not exclusively, takes the shape of a hexagon. So if we look at a drawing of its structure, we should be able to account for all of these atoms. We start naming carbons with the carbon on the rightmost point, which is carbon number 1. We circle around moving clockwise to count the other carbons, but notice that carbon number 6 is not part of the hexagon structure. It branches up off of carbon 5, which leaves the 6th point in the hexagon occupied by an oxygen atom. In most drawings within organic chemistry, assume that any corner or point where two lines meet is a carbon, if it is not otherwise labeled. Next, we can count the oxygen atoms, and we see that there are 6 total. Finally, we should have 12 hydrogen atoms. All of them are shown here, but know that on some structures, if nothing is shown to give carbon the 4 bonds it makes, we should always assume that a hydrogen is present to get it to make 4 bonds. Okay, so back to glucose. All of the atoms within our chemical formula are accounted for. Now, the last thing we need to know about glucose, which is very important, is that it comes in 2 forms, which are called alpha and beta. There is one very specific difference that exists between these forms. Take a second to see if you can spot it. If you notice a difference between the orientation of bonds coming off of the first carbon, you got it. In alpha glucose, the OH group is oriented down, and in beta glucose, the OH group is oriented up. This only applies to the hydroxyl group off of the first carbon. You can see that carbon 2 is always down, carbon 3 is up, and carbon 4 is down. An easy way to remember this is that beta follows the up-down pattern, while alpha follows the down-down, up-down pattern for the OH group. Let's take a look back at our larger polysaccharides to see why these structural components are important. Starting with starch, we can see that the chain of glucose molecules contains glycosidic bonds that connect carbon 1 of one molecule to carbon 4 of another molecule. We call this a 1,4 glycosidic bond. If you look closely, you can see that all of the oxygen atoms on the 1,4 bonds are pointed down, and all of the glucose molecules are oriented the same way. This tells us that starch molecules only contain alpha glucose, which is very important to remember. Only alpha glucose, not beta glucose. We can break this down further into different types of starch molecules based on if the structure is linear or branched. If we only have 1,4 bonds between alpha glucose, meaning the molecule is one straight line of sugars, we call this amylose. But we can see here that sometimes the structure branches, and this branch shows a bond between carbon 1 of one molecule and carbon 6 of another. And as you guessed it, we call this a 1,6 glycosidic bond. If that happens and new chains of alpha glucose branch off of an existing chain, it is no longer called amylose, and instead called amylopectin, which has a combination of 1,4 and 1,6 bonds. Next up we have glycogen, which is similar in structure to amylopectin, but it tends to branch more as it is created inside the body of an animal. This structure, because it branches, also contains a combination of 1,4 and 1,6 glycosidic bonds. And additionally, you should be able to deduce that only alpha glucose is used to create this structure, not beta glucose. And finally, we come back to cellulose. Now, the cellulose you can see is very different in structure because it is made out of beta glucose only. Because the OH group on the carbon 1 and beta glucose is up, and the OH on carbon 4 is down, when these beta glucose molecules join together, every other molecule has to be inverted, which we can see here as we count carbons counterclockwise. Every other one is upside down. This makes the cellulose structure unique for many different reasons, which we will discuss throughout the course. Moving away from carbohydrate molecules, let's now spend some time focused on lipids. We know from video 2.1 that lipids are made out of carbon, hydrogen, and oxygen, and do not follow any specific ratio like carbohydrates do. They can take on a few different shapes, but one of the main core structures of a lipid is called a fatty acid. Each fatty acid molecule starts with a carboxyl group, which has a carbon atom that is attached to an oxygen with a double bond and another oxygen with a single bond that is also attached to a hydrogen, which is also called a hydroxyl group. This is often written as COOH. From there, a fatty acid then has additional carbons attached to the first carbon. How many carbons should there be? That depends on the type of fatty acid. Some have many carbons and are quite long, while others are on the shorter side and only have a few. You will notice, though, that each carbon within the fatty acid chain has a hydrogen molecule attached to complete all 4 bonds that carbon can make. If there is a single bond formed between each carbon atom of the fatty acid chain, we call that a saturated fatty acid. You can think about it being saturated with hydrogen because single bonds means more open spots for hydrogen atoms to bond to the valence electrons that each carbon atom has. If there are any double bonds between carbon atoms, we call this an unsaturated fatty acid. Monounsaturated would have just one double bond in the chain, where polyunsaturated can have more than one. This double bond creates a bend in the molecule, which is not shown in this image, and also contains less hydrogen atoms because carbon can only make 4 bonds, each double bond counting as 2 for both of the attached carbon atoms. When we take a closer look at unsaturated fatty acids, we can distinguish between 2 different isomers, or types, based on where the hydrogen atoms are in relation to the carbons that share a double bond. If both hydrogens are oriented in the same direction as they are in this image, we call this a cis fatty acid. Cis, which sounds like sisters that are close to each other. Notice how this impacts the shape of the chain, causing it to bend. If the 2 hydrogen atoms are oriented in different directions or facing opposite sides of the molecule, we call this a trans fatty acid. This does not have the same impact on the shape of the chain compared to the cis isomer. Now that we know a bit more about fatty acids, we can start to apply some of this knowledge into real-world applications. And with fatty acid molecules, research has shown that certain types can pose risks to human health. On a nutrition label, you can find the fats within the food broken down into saturated and trans fats, with a total fat in grams and daily percent value above. The reality is that saturated fats and trans fats both pose health risks to the human body because they work to increase levels of specific molecules called lipoproteins. Now we know that fat molecules like fatty acids and cholesterol don't mix and dissolve well in water. So for the body to transport them through the blood, which contains a large amount of water, they need to be packaged to move freely. They are packaged in these structures that we call lipoproteins and act like transport vesicles and share some similarities to what we would find within a cell membrane. These lipoproteins are broken down into two categories based on how they are transporting their lipid substances, called low-density lipoproteins or high-density lipoproteins, abbreviated LDL and HDL. LDLs are used to carry cholesterol from the liver to the rest of the cells in the body, meaning it increases the amount of cholesterol in your blood, while HDLs pick up excess cholesterol in the bloodstream and bring it back to the liver for disposal. Bringing this back full circle here, when someone consumes saturated or trans fats, specific receptors that control the take-in and breakdown of LDLs are decreased while LDL production is increased, meaning a large amount of LDLs are left in the bloodstream. This excess LDL can stick to arteries and build up plaque, which can greatly constrict blood flow. This plaque buildup is called atherosclerosis, but more on that later in topic 6. Subsequently, data shows that when you consume regular cis unsaturated fats, it raises HDLs while working to remove cholesterol from the bloodstream and prevent plaque buildup. Understanding how these molecules impact the internal workings of the body is important. And lipids are only one part of a nutrition label. So much to learn. So far we have talked about lipids in terms of fatty acid chains and cholesterol. But there is another class of lipid that is extremely important to the animal body, and these lipids are called triglycerides. A triglyceride is formed when a glycerol group, which looks like this and has a chemical formula C3H8O3, binds to 3 fatty acid chains, and we already know their structure from a previous slide. The OH of each of the 3 hydroxyl ends of the glycerol undergo a condensation reaction with the carboxyl groups of the 3 fatty acid chains. This bonds them together while releasing a water molecule for each pairing, which will result in 3 new water molecules in total. This type of bond that connects a fatty acid chain to a glycerol molecule is called an ester linkage, which is a bit different than the glycosidic bond we discussed that connects sugar molecules, even though both are condensation reactions that create water. And as a side note, the condensation reactions that we are referring to here that create water molecules are also called dehydration synthesis reactions. Be prepared to see either 2 of these names on an exam. Once the bonds are created, the triglyceride is officially formed. The main function of these molecules is long-term energy storage. In animals, they manifest as fat tissue, and within plants, they create oils that share a similar purpose. Continuing from the last slide, it's important to understand the difference in how carbohydrates and lipids are used in the body in terms of energy and storage. The basics of this boil down to the fact that carbohydrates are used for short-term energy storage, as they can be very quickly transported and broken down, but the energy availability is lower compared to what lipids can store per gram. And lipids are used for long-term energy storage. They are harder for the body to break down and also harder to move around because they are insoluble, which means it takes longer for the energy to be extracted and used by cells. The trade-off here is that they can store a lot more energy per gram than carbohydrates can. When talking about lipids stored in the body, we can't overlook the concept of obesity, which is when an individual has an excess of stored fat, much more so than is within a threshold of what scientists consider to be a healthy weight. A tool that is used to measure this is called the body mass index, abbreviated as BMI. It's a simple equation that takes the mass of a person in kilograms and divides it by their height in meters squared. A value in the normal category of body mass based on height would be a BMI between 18.5 and 24.9. Going above or below this value puts you in a category that is either above or below normal weight. This tool, however, is not exact and has some faults. For example, if someone is extremely muscular, it could greatly increase their mass value based on their height and put them into an overweight category even though the measurement is supposed to be based on accumulated fat levels, not muscle. So understand how it works and use with caution.